Respiratory Physiology, Oxygen Transport in Blood. Welcome to today's episode, where we take a deep dive into the remarkable journey of oxygen through the human body, a topic central to respiratory physiology and essential for life itself. Let us begin by exploring the challenge posed by the low solubility of oxygen in plasma. Oxygen is indispensable for cellular respiration, yet its ability to dissolve directly in blood is limited. If our bodies relied solely on dissolved oxygen in plasma, the heart would need to work at an unsustainable rate to meet tissue demands. Fortunately, evolution provided a solution through the molecule known as hemoglobin, which allows blood to carry up to 70 times more oxygen than plasma alone. Now, oxygen is transported in the blood in two principal forms. First, and most substantially, it is bound chemically to hemoglobin within our red blood cells. This process is not only rapid but also reversible, forming a compound called oxyhemoglobin. Second, a very small percentage of oxygen dissolves freely in the plasma. While this fraction is minute, it plays a crucial role, as it is the dissolved oxygen that creates the partial pressure responsible for driving diffusion and determining how much oxygen hemoglobin can carry. Let us consider hemoglobin in more detail. This protein is composed of 4 polypeptide chains, each centered around a heme group. Each heme group contains one atom of iron, and each iron atom has the extraordinary ability to bind one molecule of oxygen. Therefore, a single hemoglobin molecule is capable of transporting 4 oxygen molecules. Oxygen saturation refers to the percentage of heme groups bound to oxygen at any given time. Clinicians commonly measure this parameter using a device known as a pulse oximeter. When all heme groups are occupied by oxygen, we refer to this as 100% saturation. In a healthy individual, arterial blood typically achieves a partial pressure of oxygen near 100 mm of mercury, resulting in a saturation of approximately 97 to 98%. In mixed venous blood, the partial pressure drops to 40 mm of mercury, and saturation is closer to 75%. A pivotal concept in respiratory physiology is the oxygen-hemoglobin dissociation curve, which describes the relationship between the partial pressure of oxygen and the saturation of hemoglobin. This graph is characteristically S-shaped, or sigmoidal, and offers important insights into oxygen loading in the lungs and unloading in the tissues. The upper flat portion of the curve, which represents partial pressures between 60 and 100 mm of mercury, is considered the loading zone. Here, even a significant drop in oxygen pressure does not markedly affect saturation, thus providing a valuable margin of safety. The lower steep portion, representing pressures from 10 to 60 mm of mercury, is the unloading zone. In this range, a small decrease in tissue oxygen pressure results in a substantial release of oxygen from hemoglobin, allowing actively metabolizing tissues to extract the oxygen they need. Hemoglobin's affinity for oxygen is not fixed. It adapts in response to various physiological cues to facilitate either loading or unloading as necessary. 4 major factors can decrease hemoglobin's affinity for oxygen, making it easier to release oxygen where it is needed most. These are increased temperature, increased acidity or lower pH, increased partial pressure of carbon dioxide, and elevated levels of a substance called 2,3-bisphosphoglycerate, produced by red blood cells during chronic hypoxia. This phenomenon, known as the Bohr effect, ensures that oxygen is delivered efficiently to the tissues with the highest demand. In contrast, a leftward shift in the curve, which makes hemoglobin bind oxygen more tightly, can be provoked by substances such as carbon monoxide, leading to dangerous levels of tissue hypoxia by preventing oxygen release. When discussing oxygen in the clinical setting, practitioners often focus on the total oxygen content in arterial blood, which reflects the actual volume of oxygen present. This total depends on hemoglobin concentration, saturation, and the small dissolved fraction. For example, a patient suffering from severe anemia may have normal values for partial pressure and saturation, but if the amount of hemoglobin present is low, their total oxygen carrying capacity will be significantly reduced. This underscores the importance of considering both respiratory and hematologic factors when evaluating oxygen delivery. The final step in this vital process is the delivery of oxygen from the blood to the tissues. The amount of oxygen delivered per minute is determined by 2 factors: the volume of blood pumped by the heart, known as cardiac output, and the arterial oxygen content. If either of these is compromised, whether due to low hemoglobin levels, decreased saturation, or reduced cardiac output, tissue hypoxia may ensue. In summary, oxygen transport is an extraordinarily efficient and tightly regulated process. Hemoglobin enables our blood to carry vast quantities of oxygen. The sigmoidal shape of the dissociation curve ensures optimal loading in the lungs and unloading in the tissues. Factors such as pH, carbon dioxide concentration, temperature, and 2,3-bisphosphoglycerate fine-tune oxygen delivery to match the unique metabolic demands of each tissue. Ultimately, the total oxygen content depends on hemoglobin levels, saturation, and that small but crucial dissolved fraction. The successful delivery of oxygen to our cells relies on both cardiac performance and the oxygen-carrying capacity of the blood, highlighting the intricate interplay between our respiratory and cardiovascular systems. Thank you for joining us for this discussion on oxygen transport. Be sure to subscribe for more explorations into the wonders of physiology. Until next time, keep breathing easy and stay curious. It's the Everything Surgery Podcast. Dissect the extraordinary.